Okay, so today we're going to cover chapter 4, which is an overview of the cell. It really is a tour of the cell. So what I'm going to do, I'm going to start off by talking about some of the techniques that biologists use to study cells. And then we're actually going to look at kind of an overview of the types of cells that are out there. And then we're really going to focus on something called a eukaryotic cell today. And I'm going to talk about some of the major components and what they're for, kind of what their purpose is within the cell. I'm going to end with a little video that shows a little bit of everything. But really what I want you to take away from this is to have a general idea of what is in a typical cell, and to kind of recognize the roles these different components play within the cell. So that's kind of the goal. So before we get into what's actually in a cell, I want to learn a little bit about how scientists, biologists will study cells, how we know what we know about cells. And we know what we know about cells mostly because of a laboratory technique called microscopy. And you've probably all heard of a microscope before. Hopefully some of you have used a microscope before, but microscopes are devices that allow us to view objects that are too small for us to view them with the unaided eye. So this is a kind of a diagram of scale that tells you the types of things you can see with the unaided eye versus the types of things you can see with the two kinds of microscopes that I'm going to talk about today. There are some cells that you can see without a microscope. So if you've ever had chicken, or you ever had an egg, a chicken egg, so you had eggs for breakfast, you're looking at a single cell. That is one big cell. Frog eggs, too, are things you can see without a microscope. There's a lot of eggs of organisms that are larger than most normal cells, and you can see those without the use of a microscope. But most plant and animal cells are too small, as well as the components of a cell, which is some of the stuff we're going to be seeing today, are too small to see with an unaided eye. So we'll need some kind of microscope to do that. Parts of a cell, like mitochondria and nuclei, you can see with something called a light microscope. Light microscopes are really great at seeing living cells. They're one of the most common things. If you take the introductory 1010 lab here, you use a light microscope. That's probably one that you've used if any of you have used a light microscope before. You can buy a really inexpensive light microscope from, you used to be able to get them at Radio Shack. They probably still sell them, little plastic things, and you can see stuff pretty well. They don't have the best resolution, but you can see stuff pretty well, down to the size of maybe mitochondria. Things that are smaller than that, like small bacteria, viruses, ribosomes, which we'll talk about today, some of the biomolecules we talked about on Thursday, like proteins and lipids, they are much too small to see with just a light microscope, so we use a different kind of microscope called an electron microscope. Electron microscopes have greater resolution, greater magnification. They're able to make things much larger. All right. So I'm going to show you kind of some examples of these. So microscopes, they basically work by magnifying structures. So this was one of the most early microscopes. This was by a scientist named Leeuwenhoek in 1673, so a very long time ago. All it is a screw with a pin on it and a single lens, and he would hold the lens up to the light, and he'd be able to magnify whatever was on the tip of that pin a little bit. So it's a very early, kind of not a very great microscope, very similar to like a magnifying glass would be today, where you just have a single lens and you can hold it. So if you've ever used a magnifying glass to magnify text or something like that, you can make things a certain degree, you can make them larger. You can maybe make them 10 times larger, maybe 15 times or so larger. Nowadays, we use a type of light microscope called a compound light microscope, and that's on the bottom left there. And what it does, it actually has multiple lenses. It doesn't just have a single lens. It has one up near the eyepiece, and it has one right near where the specimen is going to be housed. And so as light goes through the specimen, it gets magnified a certain amount through the first lens, and then it gets magnified again through the second amount, and we can magnify things up to maybe about 1,000 times or so with a light microscope. And that's great. So this image on the upper left here is from a light microscope. If we want to magnify things larger than that, we want to look at smaller objects or we want to get finer detail, we use something called an electron microscope. And the big difference between the light microscope and the electron microscope is what it actually shines on the specimen. So, of course I don't have markers, but so the electron microscope shines beams of electrons at the object, whereas the light microscope is light rays. An electron beam is much smaller than a beam of light, so it's able to hit smaller and smaller pieces on the object. And here are two different kinds of electron microscopes. Don't worry about the difference between them. But a scanning electron microscope does a really good job of looking at the surface of an image, of an object, and a transmission electron microscope does a really good job of looking at a cross-section of them. And by using electron beams, and then a computer, usually, that interprets the beams bouncing off the object, you can magnify an object much larger, and so you can get finer and finer details of the structures. So microscopy, whether it's a light microscope, which allows us to see living tissue, or an electron microscope that allows us to magnify things very large, they're both great tools for what we're doing here. So one of the downsides to electron microscope, well, one, they're kind of expensive to have, but also because you're getting such fine detail, you have to kill the object you're looking at, and you have to fix it. So you have to make sure it doesn't move at all. They actually will coat it in some metal, usually, which makes it very reflective to electrons. So if you want to look at living things, the light microscope's the only way to go. Okay, so most of what we're going to be looking at throughout the rest of today's lecture was learned through microscopy. So I don't expect you to be able to really read this slide at all. What this is what's called a tree of life. And what it does is it takes all living or all organisms that we know about, and it plots them based on their ancestry, basically. So things that are more closely related, things that shared a common ancestor, are closer together on the tree. So it's very much like a family tree would be for a human. So you would be really close on your family tree to your brothers and sisters, and then you wouldn't be too far away from your parents. They would be on there as well. You'd be a little further away from your cousins than you are to your brothers and sisters, and you'd be even further away from your second or third cousins, things like that. And you can keep tracing your family back until you find common ancestors. So you and your cousins would have a common ancestor in your grandparents, right? So this is what this does. It's just on a larger time scale, and it's looking at whole species and how they're related. And you can see that in the very center, all the lines come together into one point. So this is the theoretical common ancestor for all life on Earth. We have no idea what it was like. People have lots and lots of guesses, lots of hypotheses as to what that organism might have looked like, but we have no idea. It has to be something that everything that's alive now came from, so it's really hard to trace back things that far. But what we do know by looking at this tree of life is that life fits into three major groups. There's bacteria in blue, there's archaea there, the yellow and red, and then the green are the eukaryotes. Bacteria and archaea, the blue and the yellow circles there, they share a lot of common features, even though technically archaea are more closely related to eukaryote than anything else. So don't worry too much about this taxonomy part of it. We're going to talk more about this later on this semester once we get to taxonomy. For now, we're just going to learn about some of the common cell types that are found in these groups. We are in eukarya, and I don't know if you can see where we are on there. It's kind of hard to see us there. We're on the left there. It says animals. So we're kind of the bottom left of that green one. So we are eukaryotes. We're going to start off by talking about bacteria and archaea, which are prokaryotes. So bacteria and archaea are what are called prokaryotes, and then everything else is what's called a eukaryote. Prokaryotes share some common features in terms of their cells. And so we're going to start off with just a real quick introduction to prokaryotes. We're going to spend most of our time today talking about eukaryotes, which is what animals are. So prokaryotes, like bacteria and archaea, have no internal membrane system. And this might not make all that much sense right now, but once we start talking about the complexity of eukaryotes, you'll see how different this is. So no internal membrane system. So their DNA, or their genetic information, is not found in a nucleus like it will be in eukaryotes. It's not surrounded by a membrane because there's no internal membrane. It's found in a region called the nucleoid, so it is in a particular region, but it's not surrounded by a membrane. The only division in a prokaryote is the cell membrane and the cell wall, which is what surrounds the entire cell. So this is a typical bacteria cell. It has some flagella that move it forward, so those are the tails there. It's one single cell, and inside of it is some DNA, some ribosome, some cytoplasm. Talk about it. So kind of an analogy for a prokaryotic cell is it's like a studio apartment. So a studio apartment is one room, one big room where you have your bed, you have your dining room, you have your living room, you might even have your kitchen. Some really awful studio apartments have their bathroom as all part of that one room, right? But it's just one big room where everything happens. A eukaryote, on the other hand, is a multi-bedroom apartment. It's got bedrooms, it's got a living room, it's got a gym, it's got all kinds of divisions, it's got walls that will separate the different functions of the cell. So prokaryotes, no internal membrane system, no nucleus, no mitochondria, no chloroplast, none of that stuff that we're going to talk about and we're going to spend most of the day talking about. They're fairly simple compared to eukaryotes. So that's really the take-home message. Bacteria and archaea, prokaryotic cells have no internal membrane system. So eukaryotes, on the other hand, are much more complex. They have what are called membrane-bound organelles in them. They have internal membranes that divide the internal structures into units called organelles. And the whole purpose of an organelle is to regulate the activities that go on inside the cell by separating it out into different processes. You're going to have one location where energy is extracted from food. It's called your mitochondria. You have another room, another organelle in plant cells where sunlight is used to convert carbon dioxide into sugars. So you've got two different processes that are going on there. You have a location in the center, which we'll talk about, where the DNA is housed, and its sole purpose is to protect the genetic information. You have locations where protein synthesis occurs. You have locations where digestion occurs, where it breakdown, so those hydrolysis reactions we talked about the other day occur. So what we're going to do is walk through and talk about these different organelles and what their major functions are. So by the end of today, you should have a general idea of the major organelles in a typical plant and animal cell and what their functions are, what they do. Right? So DNA in a eukaryote is found housed inside of one of these organelles called the nucleus, and we'll talk a little more about the nucleus as we go forward. Whereas remember, prokaryotes, it's just floating around in the center of the cell in a region called the nucleoid. Okay. So, all of these organelles, or rooms within the cell, are surrounded by cell membrane. And this is what the cell membrane looks like. Cell membranes are what are called fluid mosaics of two different biomolecules. They're made of protein, which is the purple molecule. That's one of the components of a cell membrane. But the majority of the cell membrane is made up of the gray and yellow molecule there called a phospholipid. And a phospholipid is a lipid. So we talked about lipids on Thursday. What's one of the main features of lipids? Okay, fats are a lipid, but what's like one of the characteristics of a lipid? How is a lipid different than the other biomolecules we talked about? So we talked about carbohydrates, we talked about proteins, we talked about nucleic acids. What was real largely important about lipids?
They only have two fatty acid tails.
Okay, well, fatty acid tails are a major component of lipids, and yeah, fatty acid tails make lipids nonpolar. So because of those fatty acid tails, they're nonpolar. So phospholipids are kind of pretty special in that they are two fatty acid tails. I don't know why I forgot my markers today, but there are two fatty acid tails and one phosphate group, and the phosphate group is polar. So you've got the two yellow parts to it on this graphic are nonpolar, and the gray part there is polar. So you've got a polar region and you've got a nonpolar region. This is why it arranges like this. So the nonpolar, the yellow parts of it, will face each other. They're towards the inside. And the gray part, the polar region of the phospholipid, faces out towards the fluid on either side of the membrane. So this creates a fantastic barrier to things passing through it. So anything that has a charge on it is going to be attracted to the polar head of the phospholipid. And it won't be able to actually get into the internal part of this membrane. So as soon as it would try to go into the internal part of the membrane, it would be turned around. It would be pushed back out to the outside of the membrane, right? So this prevents anything with a charge. So any polar substance, any ion, anything from quickly going through the membrane. And we'll eventually talk a little bit more about the proteins and how proteins embedded in the membrane will allow things to pass through it. But it provides a very good barrier to things moving across it. And some of the terms you'll see in your textbook are hydrophobic and hydrophilic. And the phospholipids' tails, the yellow part there, are what are called hydrophobic. Doesn't really mean that they're scared of water. It just means that they don't mix with water. They separate from water. So they're nonpolar. And then the head is hydrophilic, which means hydrophilic actually literally means water-loving. But in this case, it means it will dissolve in water. It will mix with water. Okay. So membranes are made out of phospholipids and proteins. And the phospholipids create this bilayer that prevents substances from moving across it. Okay. All right. So one of the most predominant structures that you'll see in a cell is the nucleus. And this is one of the most recognizable parts of a cell. It's usually fairly large. If you were actually to scrape out your cheek cells and look at them under a microscope, you could see the nucleus inside of them. It's pretty much the only thing you could make out. If you do take 1010 lab, that's one of the things you do. You scrape the insides of your cheek and you look at your cheek cells. It's not that gross. So you can see a nucleus. A nucleus is a large central plasma-bound structure, and what it does is it contains the DNA, which is the blueprint for life. And we'll talk more about DNA in a bit, but DNA is the instruction manual for how to make proteins, and proteins control a lot of what your cell does. So protecting that instruction manual is pretty important, and your cells all have a little room called the nucleus where they do that. The nucleus, as you can see here, has pores, it has openings, and it can actually open and close those pores to allow things in and out. So it'll want to allow some of the instructions to leave. It'll actually allow copies of the instructions to leave, which is RNA, and it'll allow molecules to enter that will regulate which recipes are actually being read at the time. So that's the nucleus. It's in the center. It is the location of DNA, which is a blueprint for life. Okay. Just outside of the nucleus, this kind of folded region here, is something called the endoplasmic reticulum. The endoplasmic reticulum is a large, large membranous body. It's very folded. It's where most of the membrane in a cell is found. This is actually a location for membrane synthesis. So that's where I say the manufacturing site for molecules. The lipids that it makes, one of the lipids that it makes is phospholipid. It actually will make membrane, and it can then send that membrane out into the cell. So let's say your cell needs to grow, needs to get larger. It needs to actually add phospholipids to the outer cell membrane. And to do that, it manufactures them here, and it sends them out to the outside of the cell and makes the cell a little bit larger. So that's one of the roles of this. But one of the major roles, and something we'll really focus on, is it's a site for protein synthesis. All those little dots are something called a ribosome, and we're going to talk about that in a few slides, I believe. Just double check here. No, we should eventually. So all those little dots that are on the ER are ribosomes. Those are structures that will read RNA, which is the message or the instruction manual for how to make proteins that was copied from DNA, and it'll make proteins. So ribosomes actually manufacture proteins. So, and they often do that on the ER. They're attached to the ER at the time. Okay. So here's kind of a closer look at the ER and what's going on the surface of the ER. So the blue thing here is a ribosome, and the little purple dots that are going into the big purple structure is a protein. So a ribosome manufactures a protein, and it sometimes will manufacture it into the ER, within that folded membrane. And then what happens is a bit of that membrane pinches off around that protein to create something called a transport vesicle. And that transport vesicle can then transport that protein to other parts of the cell or to the outside of the cell. So because you're manufacturing phospholipids as well as proteins here, you can create these little capsules and send them out to other parts of the cell. So it's like putting something in a box and then mailing it. The proteins are wrapped in a membrane vesicle and then sent off. All right, so that's the ER, site of protein synthesis, site of lipid synthesis. Okay. One of the locations that the ER sends its material to is the Golgi apparatus. So the Golgi apparatus is another membrane-bound structure, or membranous structure on the outside, or in the inside of the cell. And the Golgi is a site for receiving, refining, storing, and distributing chemical products. So those proteins that just budded off of the ER, that purple vesicle there, will come to the Golgi, and they'll sometimes be modified. And by modified, they might have things added to them. They might have carbohydrates added to the protein. They might have parts of the protein chopped off. They may be combined with other proteins or other molecules. And then they might be repackaged into a different vesicle and sent in a different direction. So distribution is a big aspect of the Golgi apparatus. It can decide to send vesicles to the cell surface. It can decide to send vesicles to the mitochondria or the chloroplast, which we'll talk about in a minute. It can send those vesicles wherever they might be needed. So that's the Golgi apparatus. Okay. So now we've seen most of the major protein kind of chain for the cell. The message for how to make a protein starts in the nucleus. The protein is made in the ER. It's then sent to the Golgi to be repackaged and then sometimes sent to the cell surface. So that's one kind of train of where proteins will go in a cell. other functions a cell might have besides protein synthesis and protein management is that cells need to be able to break things down. So they have these structures in them called lysosomes, which are digestive compartments. So particles that come into the cell will often first merge with the lysosome to break it down. So let's say you've just eaten lunch and you process of digestion, you first chew your food and you break it into smaller pieces. You then swallow it and it gets broken down more in your stomach by all the acids and different enzymes that are in your stomach will break it down into smaller particles. It'll get absorbed by your bloodstream, and then from your bloodstream, it will enter cells. And it's still a pretty large molecule. It's not broken down all the way to a monosaccharide. It's still a polysaccharide or an oligosaccharide. It's a larger molecule. So it will be merged with something called a lysosome, which contains what are called hydrolytic enzymes that perform hydrolysis to break that large biomolecule down into smaller biomolecules. And that's the process of digestion. You now might have monosaccharides or disaccharides, which can then head to the mitochondria and be broken down further to extract their energy. So a lysosome contains enzymes, contains molecules that will break down food particles. They also function to recycle damaged organelles. So if you have something that's not functioning properly inside your cell, rather than just getting rid of it, your cells will break it down into its components and use it to build something new. So it's like taking apart a wooden box to build a new wooden box. Like that. You're going to recycle the components. Lysosomes do that as well. So lysosomes are a fantastic kind of structure within a eukaryotic cell because they're a very specialized sack of pretty horrible enzymes. And just imagine if those enzymes and molecules were just floating around freely inside the cell, they might be just randomly digesting and breaking down different components, and the cell wouldn't be able to function. But because the eukaryote has all this membrane and can create these little vesicles, they can put all those horrible enzymes inside of a little vesicle called a lysosome and allow it to digest things by just merging with a sac containing whatever you want to digest. So a pretty cool little process. Okay. There are also some larger sacs of membrane inside of a cell with a variety of different functions, and pretty much a generic term for a large vesicle or a large sac of membrane inside the cell is called a vacuole. Vacuoles are a site that can store some cell products that are not needed. They're kind of like a storage closet, basically, in the cell, and they can just store things there. In plants, a lot of the center of the cell is taken up by what's called a central vacuole, a large central vacuole. And one of the things that stores is it stores water. There's a lot of water in the center of a plant cell. And because of that vacuole filling up with water, it pushes outward on the edges of the plant cell and makes it solid. So you've probably noticed if you've had plants that if you don't water them for a while, they get really droopy and floppy. Right? That's because the vacuole, when it loses water, it stops pushing on the outside of the cell, and the cells start collapsing on themselves. So by watering your plant, you're filling up the central vacuole of all the plants, and it's able to give it that upright, more perky look to the plant. Right? So there's lots of different roles of vacuoles, but they are just large sacs of membrane. They can be storage. Some of them are where they take toxins. Some cells will store toxins in these for later, to get rid of those toxins later on. And then, again, in the plant cells, there'll be central vacuoles. In some single-celled organisms, they're actually able to move that vacuole, to contract it. It's called a contractile vacuole, and that will actually help it in movement and in defense sometimes. So by moving that vacuole, it'll fill it up with water, and then it will quickly empty it of water, and that allows it to to move and defend itself. Okay. All right. Two other very prominent organelles, which we're going to have an entire chapter on each one of these, are mitochondria and chloroplasts. And these are also, both of these are found in plant cells. In animal cells like us, we don't have chloroplasts, but we do have mitochondria. These are sites of energy conversion. And we're going to have entire chapters on these, so I won't go into a lot of detail about them now. Mitochondria convert chemical energy in food into cellular energy, something called ATP, which we're going to talk about in a few chapters. So it converts one form of energy to another. Chloroplasts take energy from sunlight and convert it into food energy. So in plants, they use both of them. They take sunlight energy, and from their chloroplasts, they use that sunlight energy to make sugars, and then they send the sugars over to the mitochondria and convert that sugar energy into ATP, which is the energy that will drive lots of processes inside the cell, like movement of those vacuoles, movement of those vesicles, creation of proteins. In animals, we use that energy to move our muscles, to pump our heart, things like that, to move things around. So mitochondria and chloroplasts, sites of energy conversion in plants and animals. Okay. Let's see how I'm doing here. Okay. I'm rocketing through this pretty fast, so stop me if you want to go over stuff again. Well, I'll go back through a lot of these and review it again. The final thing I want you to know about cells is a little bit about something called the cytoskeleton. And this is actually a very cool thing inside of cells that we haven't actually known for all that long about it. We didn't talk about it all that much when I was an undergrad. We're talking about it more nowadays. It's a network of protein fibers that provide a structural framework inside of the cell. So I'm going to show you a video in a minute or two that shows you kind of an overview of a cell. It starts from the outside of the cell, it works its way in, it looks at a lot of these structures we've been talking about, and then it works its way back out. And one of the most prominent things you'll see is it looks like there's this lattice or struts that are everywhere in the cell. This is the cytoskeleton. So here, this is an image that has highlighted a portion of the cytoskeleton called microtubules inside the cell, and you can see that they radiate from the center of the cell all the way to the outside. So cells are not just blobs. They're not just sacs. They're not like water balloons that just has fluid in the center and just a membrane on the outside. Instead, they are highly organized, and the shape of the cell is due to the structure of the cytoskeleton. So if the cytoskeleton can create these very elaborate structures. So for example, if you look at the intestinal cells so the cells in your intestine, the surface of the cell is really, really highly invaginated. It has lots and lots of folds to it. And this allows, as food goes across those cell surfaces, it allows a lot of surface area for absorption of nutrients. Without those folds in your intestinal cell membrane, you wouldn't be able to digest food very well at all. And there are people that have conditions where they lose that structure in their intestinal cell wall, and they have a hard time digesting things properly. They have a lot of GI problems. So there's a lot of things, especially if they get swollen or inflamed. There's a lot of bad conditions that can result from that. The reason why you can have that folding in the cell surface is because there are cytoskeletal elements holding them up. So some of the things cytoskeletons do, one, that structural support is what I was just talking about. They're also a transportation system. So you'll see this in the little video, that there are these large cytoskeletal elements, And that's what the vesicles move along. Vesicles are these sacs of membrane. They don't just float inside of your cell. They're actually transported inside your cell. They have these little motor proteins that hold onto them, and they walk along the cytoskeleton, dragging the vesicle along with it. So structural support, transportation system, guiding cell division. So later on this semester we'll talk about mitosis. We'll talk about how cells actually divide. And this is all guided by these large structures that are inside the cell, the cytoskeleton. Cell connections. So your neighboring cells are held together by cytoskeletal elements. So your muscle cells are held to your bone cells by something called a desmosome. What it is, it's a cytoskeleton filaments that stitch together your muscle cells and your bone cells. And if you tear a muscle, what you're doing is you're breaking some of those junctions. Very, very painful thing. It takes a long time to heal because you've got to reform those junctions between neighboring cells. You also have cell connections between the cells in your intestine that are what are called, they are these watertight junctions between the cells, and they prevent fluid from going between the cells. If you break those, some of your fluid from the inside of your stomach leaks out of your stomach, and you have what's called an ulcer. So you're breaking those what are called tight junctions. They're junctions between neighboring cells. So you have lots of different types of cell division or cell attachments that are all made by different cytoskeletal elements. And then one of the final things it does is enables movement. So you'll see this in the video. The outside of a cell surface is covered in cytoskeletal elements, and they allow your cells to attach. And then by changing the cell shape, you can actually cause a cell to move. All right. So what I'm going to do, I'm going to show you a little video, which is something I will post it on Canvas so you can see it again. It's an overview of a lot of the different parts of the cell. I'm going to narrate through it and point out the different parts, and then we're going to go back and look through some of these things and just make sure we understand all the main cellular components and what their roles are. But that's basically all this chapter is an overview of all those different parts and some of the main functions of all of them. Right? So here's a video. This was done by a group at Harvard, and it's a pretty cool little video. Let's see if I can get it to work. So it has a neat little music background, but I will talk about it. Okay. So this is, you're in a blood vessel, and what it's going to do is going to start from the outside of a cell and work its way in. So this is what's called the extracellular matrix. This is cytoskeletal elements on the outside of a cell which allow for cell attachment. Here's the surface. These are those phospholipid heads. You can see they're very movable, and there are lots of proteins that stick on both sides of them. So now we're on the inside of the cell, starting to work our way down, and you're going to see more of these cytoskeletal elements. So here's this kind of grid work of cytoskeletal elements that support the cell's shape. And these cytoskeletal elements are not fixed. They can actually be moved by the cell. So your cell can change its shape. It can also create new transportation pathways just by rearranging some of these proteins that make up the cytoskeleton. This is something called a microtubule. This becomes kind of like intercellular highways. So it can build them and break them down. And here is a vesicle being moved by a motor protein. So super cool. These giant vesicles, and they actually literally walk along these microtubules, pulling the vesicles with them. So here we're looking towards the center of the cell. There's the nucleus, there's a mitochondria. This is going to zoom in on the nucleus. And here's RNA coming out of nuclear pores. RNA is going to meet up with a ribosome, which is what's going to start making a protein. And so here's a ribosome attaching and beginning to make a protein. So the yellow thing is a protein. And one of the places it can make a protein is in the endoplasmic reticulum. So you're going to see this ribosome, soon you'll see a ribosome attached to the endoplasmic reticulum. And now it's making a protein into the ER, which will be again, folded into a vesicle, will bud off the ER, again, be transported by these motor proteins. And one of the locations it can go to is this. What is this? Remember what that's called? Your Golgi apparatus, right? So that's the Golgi. And then proteins can be excreted from the Golgi, sent out to anywhere else, so outside of the cell. So again, here's the cell surface. Here's some proteins on the cell surface. And one of the things they can do, the proteins can attach to neighboring cells to communicate with them, to attach to them, and like those, like your muscle cells attaching to your bone, or other kinds of attachment. Now let's zoom out again. All right, there we go.